Why don't you come and grab a seat and get comfortable, because I hope you're going to stay here for the rest of the afternoon and listen to what promises to be an inspirational and exciting and pretty phenomenal alumni lecture series. No pressure, guys. I'm Lee Sublett. I'm from Alumni Affairs, and it's my absolute pleasure to welcome you to the annual WEP Alumni Lecture Series. I think this year is pretty exciting because we've got four very distinguished alumni presenting on topics that have a lot of relevance to the topic of personalised medicine. And along the way of them talking about the research that they're doing, they're also going to be talking a little bit about their journey since they left KAUST. So not only is it a session that's going to be focused on the research and the topic of WEP, but it's also, I hope, with you as students, going to give you a sense of what our alumni go on to do after they leave KAUST. And although KAUST's alumni community of 1,990 is small in size, the individuals who make up that community are making an enormous global impact. And it's my belief that you're privileged today to have the opportunity of hearing from four of these inspirational alumni. Again, guys, no pressure. Allow me to introduce by name your speakers for the afternoon. And the first one who will kick off in just a moment is Demetrius Collector. He'll tell you a little bit about where he works now and the research that he's undertaking, but he graduated from KAUST with his PhD back in 2016. We have Faisal Nawab, who graduated from KAUST in 2012 and is amongst our 325 KAUST founding class, and he completed his MS in 2012. We've got Eduardo Gorin, who graduated with his MS in 2012. And we had Awa Reis, who some of you might know because she's a relatively recent graduate, having completed her PhD in 2018. So I know some of you come with the intent of getting your ID scanned and with the intention of just doing that and then wandering out. It would be my advice, given that we've got four alumni who have come back from all parts of the globe, for you to sit back and enjoy the rest of the afternoon, because these are four of the best undertaking research in some of the world's best institutions and organisations. And I really do think that their presentation this afternoon will inspire and hopefully be that little bit of a motivation should you be feeling a little bit kind of down in your studies at the moment. So enjoy the afternoon, please don't rush away, and allow me to invite the first presenter up to get organised to talk to you. Dimi, thank you.
Hello everyone, it's my great pleasure and honor to be an invited speaker to this alumni lecture series for WEP2020. My name is Demetrius Collector, and today I'm going to talk to you about liquid biopsies and the application in personalized cancer medicine. So first, few words about my background. I'm originally trained as a computer engineer. I moved to KAUST in 2011, where I completed my PhD in 2016 under the supervision of Professor Panos Kalnis. And then I moved to cancer research. I had placements in different countries, different cancer institutions. Now I'm affiliated with the University of Bergen, and my specialization is on the analysis of cancer genomic data with applications in personalized cancer medicine. Of course, I have a long experience in bioinformatics, and I have developed several tools. So today, I'm going to first give you an introduction about cancer and some principles that we apply in precision cancer medicine. Then we will define liquid biopsies and how we can use them, what are the potentials and the limitations. And at the end, I will give you some results. I will present some results from my most recent work. So it's not difficult to convince you about the consequences of cancer for the life of people and the families and friends. And for this reason, cancer is not just a disease. It's more than that. There are several types of cancer. Here I have some statistics. So, for instance, breast cancer is the most frequent forms in women, followed by lung cancer, prostate cancer in men, and different other types of cancer. And all of the cancers are characterized by an uncontrolled growth of abnormal cells, as you can see here in this figure, where the cells actually do not die, they do not go to cell death by apoptosis. Of course, the disease is different. Although the cancers are alike, the disease is different. And actually, the individual's patient disease is different. And this makes a good link to precision medicine. Because we have different disease, different individuals, we need to develop different drugs. And what I described you in very simple words was actually the topic of a very important paper in cancer research, which is entitled The Hallmarks of Cancer by Hanahan and Weinberg in 2000. They defined some functional capabilities of the tumor cells to grow, proliferate, and spread that involve, for instance, alterations in the signaling, alterations in the microenvironment to provide nutrients, the ability to escape death, and metastasize. This paper is very important because it changed the way we study cancer and the way we treat cancer. In 2012, it was, let's say, an updated version of this work, where they introduced the so-called cancer-enabling characteristics. And I would like you to pay attention on the following. So they talked about the immune system, because there are alterations in the immune system do not kill actually the tumor cells, but they promote the tumor cells to grow. And the most important is the instability, genomic instability. And we're talking about mutations. Now we know that all cancers are mutated. And just to remind you, I'm pretty sure that you already faced that during this program, that all of us, we have mutations that are called germline mutations, that are actually, we inherit from our parents. But cancer introduces more throughout the process, the tumorigenic process. And these are somatic mutations. And we can have single nucleotide substitutions, just one position in the genome changes, but more complex things, like, for instance, deletions of chromosomes or parts of chromosomes, or amplifications, or even combination of the different things that we result to translocations. The important thing is that we can characterize the tumor genomes based on these mutations. And the reason that we know now all of this is because of tumor sequencing. And I will talk about in the next couple of slides. So thanks to the technology, tumor DNA sequencing allows us to profile patients. And what is happening in reality is that usually the patients, they go to the clinic, they have a surgery, so they extract some tumor material that then we take to the lab. DNA is isolated from the tumor material, and we can actually see, we can sequence the tumor genome. This is very important, and it's very fundamental in precision cancer medicine to characterize the tumor genomes. And here I have some numbers from the, let's say, from the beginning of sequencing. It was in 70s. This is Fred Sanger, who won the Nobel Prize for sequencing. And you will appreciate by these numbers that sequencing back in time, before 2000, was the release of the Human Genome Project, was a very laborious process. So here, just to make it more easy for you, I report how many bases was able a very good technician to sequence during different periods of the time. And you will see that it was not possible to sequence a genome before 2000 in practice. So we cannot discuss about medicine, of precision cancer medicine, before 2000. And we are privileged, actually, that we have all of this technology that allow us to have precision cancer medicine. To obtain this, we're talking about the DNA sequencing revolution. It means that the technology changes. And here, on the left, we have the equipment of around 2000, and the cost was huge, and the process was still slow. And on the other side, we have the new machines that can sequence a genome within a few hours. So we have good sequencing, and we can take benefit out of it. And also, the good news is that the cost is dropping. So only a few thousand. We need a few thousands to sequence a genome. And this is a big improvement. Let's see now how we can apply this advantage to precision medicine. Let us assume that we have this cohort of patients that are different. We have sequenced them, we have profiled them, and we know the mutations they have. What can we do now? One approach is to give them the same drug, the same chemotherapy, for instance. But we know that this does not work very well. A better approach which actually is linked to the current engine of precision medicine in cancer, is to characterize them, so generate some groups, let's say, and then in different groups give the appropriate drug, or combination of drugs, or modify the dose of the drug. This, of course, is a very big improvement, but does it mean that we have solved the problem of cancer? How did you have the best, let's say, way to treat cancer? The answer is no, and there are some concepts. The concept is that tumor evolves in space and time, which means that the tumor genome changes very, very quickly. And for instance, here I have highlighted this work from my colleagues in London, where what they did, they sequenced individuals with cancer, colorectal cancer, for instance, and they sequenced the primary tumor and the metastasis. But not only the metastasis, they sequenced different parts of the metastasis. And they found differences, which means that the sampling, when we want to characterize the genome, the way we sample the data in order to understand what is going on matters very, very well. So now, let me introduce what we are doing in practice. So this framework actually give you an abstract of the current precision cancer medicine engine, which means that in the beginning, usually in clinical trial or during the design of a drug, we have some sequencing data from a cancer patient. Then we apply sequencing or other omics technologies to characterize. Then we make some reports. We collaborate with a medical doctor. The medical doctor makes some decision about the therapy. And then we're waiting till we have a clinical response. Usually there is a metastasis. And the patients, they go back to the clinic, and this process is repeated till we have actually some resistance to the therapy, and it's not difficult to make something to save the life of this patient. And actually, this has been reported that most of the tumor material are never materialized because it's painful, it's costly, it's not practical, and they have been reported accidents during surgeries or during needle biopsies, where the needle biopsies break and they spread the tumor cells in the body. So this, although it's an improvement compared to, let's say, the one-drug-fits-all approach, still has limitations. And this makes a link to liquid biopsies. And in very simple words, what is a liquid biopsy? Is the analysis of tumor material obtained using a minimally invasive way through blood or other body fluids, for instance, urine. And this goes back in time that we know that cell-free fragments of DNA circulate in the bloodstream. In patients with cancer, some fraction of this circulating DNA originates from tumor cells, and it's called circulating tumor DNA. ctDNA is the acronym. And this goes via apoptosis, necrosis, or other biological processes, or maybe it's from the tumor microenvironment. Out of this, we know actually that we can characterize the tumor using this source of information. And this is called liquid biopsy. So now let's together see how we can apply this concept to apply the previous framework of precision cancer medicine that I presented you. Let us assume that we have this individual, this cancer patient, that has this longitudinal cancer journey. Has surgery, then develops a metastasis, the doctor gave a drug, and then we have a drug resistance. This is typical in some cancer patients. What can we do? Before, it was necessary to do surgeries, to do operations here, to do operations here, and maybe here. It's very, very difficult. But now, we can start collecting blood. And because we can characterize the tumor, we can apply DNA sequencing, epigenomics, transcriptomics, proteomics, different technologies that we know very well how to do. And we can have sequential time points. We can, for instance, ask the person to come every 2 months in the clinic, and we can collect data. So out of this point, we will have a very good readout of the disease before the metastasis. But even if the metastasis is there, after that, we can monitor, in fact, the responses to the treatment. And this is a very big advantage. Let's say if this cancer patient has a journey of 5 years, at the end, we will collect big data, we can make trajectories of the disease, and we can apply big data analytics and potentially make models to predict and save the life of other people. If we're talking about this individual, also maybe to have more capabilities to provide the right drug, the right moment. And this drug will be tailored for this particular individual because we know very well his genome and how the genome evolves in time. In this slide, I have a comparison of what I explained to you before. We're talking about here, there are also other analytes to monitor in the blood, like circulating tumor cells or RNA, but let's focus on ctDNA, which is a DNA fragment. And here is the comparison with a tissue, with a solid material that we extract using the surgery. There are many advantages. For instance, that we can monitor clonal evolution, find the resistant mechanisms, and even predict early the disease. Of course, it would have been impossible to do this type of work without appropriate technologies. Thanks to the sequencing revolution that I told you before, we have now the appropriate technologies. And we have two types of technologies. We have the low throughput and the high throughput. The low throughput are conventional genetic techniques, like, for instance, the digital droplet PCR, where we can monitor few positions in the genome, few mutations. Of course, this type of analysis is cheap, few hundreds of dollars, and very fast. And it's also very accurate, meaning that we can detect very rare fragments, mutated fragments, cancer fragments in the blood. Of course, thanks to sequencing, we have these approaches. And with targeted sequencing, we mean that we can design a panel of genes, we can sequence it in a high depth, and we can find more mutations. We can monitor very well the disease. Of course, if the budget allows, we can sequence the full genome. And by sequencing the full genome, we can identify all possible mutations, structural variants, or all the mutations of this individual, and have a better estimation of the disease, of the tumor burden. Of course, there are challenges in this type of analysis. And here, just to make it easier, I have two types of limitations, ones that come from biology, and more technical ones that I'm going to talk to you more, because it's more into bioinformatics, which is my specialty. In terms of biology, in terms of knowledge, we don't really know what is the composition of nucleic acids in healthy individuals. So we need to do work on that to find, for instance, the origins in this composition. An example is that if, let's say, lung cancer releases more DNA compared to the liver, we need to answer these basic questions. Something which is related more to tumor biology, we don't know to what extent these liquid biopsies reflect the heterogeneity of tumor, or what are the factors, features, the biological features that influence this release of mutated fragments. For instance, is a big tumor releases more DNA than a small tumor? Does the location matter? We need to answer this type of questions. Also, we don't know if all cancers can be monitored in the same way. Are aggressive tumors can be monitored better than non-aggressive tumors? So we need to do basic research on that. And now, I'm going to talk more about the technical problems, problems that we can solve using bioinformatics. First one is how to combine different analytes. For instance, there is no pipeline, if you want, or optimal strategy to integrate this data and do a multi-omics analysis that includes DNA, RNA, proteins, metabolites, all of these type of things. And I would like to talk more about the technical difficulties. So now, we need to, let's say, develop a blood test, and we want to apply this test in the clinic. This means that we need to be very accurate. We need to have very high sensitivity and specificity. And you need to keep in mind that the type of data we are dealing with are noisy. Imagine that you want to track some fragments of a cancer in a very early stage, and this is very rare fragments, so you need to be very specific. Of course, the technology is not perfect. There are errors introduced by the technology that we need to overcome these errors. And of course, the tumors are heterogeneous, so imagine that you have different metastases that release different mutated alleles in the bloodstream. How we can distinguish these things? So there are technical limitations, but thanks to the technology and to the bioinformatics research, there are solutions. And I will start with some commercial solutions. I'm not advertising any company, but it's good to let the people know what is going on. So there are actually FDA-approved tests, blood tests. One is called COBAS, this one, and the other is called TheraScreen by Roche and QIAGEN, that they apply digital droplet PCR to track specific mutations that are related to some targeted therapy. Of course, there are companies as well. Here are some examples that they offer liquid biopsy programs. And right now, for instance, they collaborate with institutes and hospitals, and they're running the Circulating Cell-Free Genome Atlas. So there are improvements. But if we're talking about academic work, there are hundreds of published papers on liquid biopsies. Here are some examples. If someone is interested, I can give more literature. And I would like to highlight a few of them. Like this one, for instance, which was one of the first one that applied targeted sequencing to detect early cancers. The method is called TechSeq. Or this one, which is actually similar approach. It's called CapSeq. This is the experimental protocol. And also they developed an algorithm, a bioinformatics algorithm, that takes this data and removes the errors, so increases the sensitivity and specificity of this, of the detection of mutations. And this makes a link to my contribution. So I will try to present you some results of my work. And first, I'm going to present the development of variant caller. For those that are not familiar, variant calling is when we have sequencing data and we want to identify the mutations. This is a difficult problem in bioinformatics. And it's much more difficult when we're talking about cancer, and even more difficult when we talk about circulating tumor DNA for the limitations that I explained to you before. So I will present you this development. It's a program which is called AmpliSolve. Then I give you a more complete piece of work, which is actually the development of a blood test together with bioinformatics analysis to suppress the errors and identify variants with very high accuracy, which is called Duplex Collar. And I have also explored other avenues for biomarkers discovery. In breast cancer, I have a study in breast cancer, but I don't have time to present all of these results. So let's begin. So the first work, this variant caller, is attached to this study, and it is a joint project with University College London. The colleagues, they had collected a big cohort, around 500 samples from castration-resistant prostate cancer patients that were sequenced using this technology, which is called ion torrent. Ion torrent is a little bit obsolete technology. They had initiated this study 5 years ago. The good thing with this technology is that it's cheap and fast, but the sequencing is more prone to errors. So the variant calling is a big problem. So we wanted to do something better. So we started analyzing this data, and we observed that actually this technology has position-specific and nucleotide-specific errors. As you can see here I have highlighted the error levels that our algorithm computes using in consecutive positions in the genome. This is in chromosome X, androgen receptor is the gene. And you will appreciate that, for instance, cytosine makes more errors in sequencing. So now the idea was to develop, based on this observation, to develop a bioinformatics method, which is maybe inspired from supervised learning, for those that are familiar, to learn from these errors and predict variants with better accuracy. And we developed this variant caller, which is called AmpliSolve. Just not many details. So it works in 4 steps. The first, we learn from the control data. So we need some control data to learn the error levels. We built a dictionary of the errors. And when you give us tumor data to screen, then we have this error, the database of errors. We apply some statistical modeling. Promise no formulas on that. So we apply some statistical modeling, and then we can decide if this is a real variant or not. So these are good news. So we developed this method. We achieved good trade-off for this particular technology. We were able to detect variants at approximately 1% frequency. And most importantly, it's a free bioinformatics software, so you can download it from my webpage. And if you are interested, you can reproduce, you can use it. So now the question is that, can we do something better? 1% is okay, but now we want to go even lower in terms of frequency, detect even more rarer mutated fragments. And the answer is yes, we can do something better, but we need a new technology. And this is called error-corrected circulating free DNA sequencing with molecular identifiers. The simple words is that When we have the DNA, imagine this is the double-stranded DNA, we just put some barcodes in the DNA. And when we sequence, and when we have the amplification of the DNA, which is the most, let's say, error-prone step, we know we can identify the mistakes. For instance, let us imagine that all of these reads, they have the same barcode. So we know that they come from one particular molecule. But this one has this mutation, which is the dot with the green. But his brothers here, they don't have it. So we can find the error. And we can say, OK, this is not a mutation. It's a real variant. Sorry, this is not a real variant. It's a mistake. Of course, the opposite is here, when the real variant is supported by all the reads. So this is more experimental, but this has many potentials to do bioinformatics analysis. So based on this idea, in collaboration with Institute of Cancer Research in London, we developed a blood test, which is suitable for colorectal cancer. The panel, we screened 32 genes. The experimental protocol is optimized using these barcodes that I described. Can be applied in the clinic with only 30 mL of blood that the doctor needs to take. And from my side, I developed the bioinformatics software to process this data. And we also developed a tool which is kind of unique. And this tool is called Duplex Collar. It's, let's say, a post-filtering bioinformatics method that can identify which variants are real and which variants are not. Can be applied in other blood tests, for instance, that they use maybe a different experimental protocol, but the data look similar with a similar configuration of the barcodes. And the idea is very simple, because it is an in silico way to identify which mutations come from double-stranded DNA molecules, which are actually the proxy of tumor cells. So we achieve very good results with that. And to convince you a little bit more, here are some data from a clinical trial. So we have 28 individuals with colorectal cancer. And the other axis, we have cancer genes. We screen them using this assay, the plasma. And with green color, we highlight the mutations that we found that were concordant between sequencing of the actual tumor obtained with surgery and the blood. We found very good concordance. Of course, in plasma, we found more mutations. And most of them we validated using more sensitive approaches, and they were real. This highlights actually this heterogeneity that I described before. And the other way around. So there were many cases where we couldn't find a mutation in the blood, but the mutation was present in the solid tumor. And this actually is expected. And this also highlights the limitations to track very rare cancer fragments. And just to convince you a little bit more, we didn't have any evidence, actually. There was no mutated read in the ctDNA sample to have this support these mutations that were observed in the solid tumor. Pretty much, this is the description of this tool. We managed to reduce the false positive rate by 97.5%. We applied this to this clinical trial, and actually now I'm collaborating with ICR, and we're applying other data from other trials to track driver mutations in some cancer genes. Again, the software is available, so you can download it. And actually, it's a nice story, because some people from Brazil, they were interested. I didn't know them. So they contacted me. I helped them with the analysis. They offered me some co-authorship. And with that, pretty much, I'm reaching the end of my talk. I hope that I convince you about the potentials of liquid biopsies. I believe that the future of precision medicine is in liquid biopsies, and most importantly, that we can use them for patients' benefit. Of course, we can use them in a research setting, not only in a clinical setting, to understand cancer biology. And it has been impossible to do all of these improvements without bioinformatics. So we need computational tools and bioinformatics. And I'm reaching the end. I would like to thank, first of all of my colleagues, especially Marco Gerlinger from ICR, Gert Attard from UCL, and Ian Tan from Genomics of Singapore. I would like to thank the WEP committee for inviting us. It's really nice to be back in KAUST. And I have one last slide that I would like to share with you, and I would like to say a few words in memory of Professor Vlad, who was, when we talk about bioinformatics, he was a colossal figure in KAUST, and was the supervisor and the mentor for many alumni. With that, thank you very much. Do we have time for questions? If there is any question, I will be happy. Otherwise, you can reach me after the talk. Okay. Oh, yeah. Do I have to throw you the Microsoft?
As a clinical tool, what do we expect the cost per patient to be for, let's say, a monthly liquid biopsy? Is this something that's going to, be a major shift in the way, cancer patients are treated financially?
It's expensive.
Yeah.
Many thousands of dollars. Yeah. Per sequencing.
Is that the sort of the clinical time, or is that the computer processing time?
The computer processing time is not the problem. We have cloud computing, we have parallel computing who can do very well. The cost is the sequencing itself, the reagents and the experimental thing. Most of the trials right now are supported by some pharmaceutical companies. And the people that are enrolled in the trials, maybe they have some agreements. I don't know all of these details. But I know for sure that many cancer hospitals collect blood in order to use it when the sequencing becomes cheaper. So it's the future. And sequencing becomes cheaper and cheaper. And we hope to reach a level where we can sequence just for a few hundreds of dollars, and it will be affordable for everyone.
Thank you.
You're welcome.
What would be the reproducibility result of this experiment? In other words, can you reproduce the result, or only you take one sample, put into the machines, and get the result, publish the paper? Can you reproduce the result, or the result was reproducible?
The results are reproducible, of course. If you mean the variant calling, or, I mean, if you, for instance, I give you the same BAM files, the same actual clinical data, you can follow the pipeline. Most of the mutations, if you are talking about the big table that I presented you, most of these mutations were validated using orthogonal approaches. So we do the sequencing, but to validate if something is not, we don't have, let's say, we don't believe that, we validate it using an orthogonal approach, like digital droplet PCR, or we sequence the actual tumor, and we can find the same findings. In this, I don't know if I answered your concern.
Doesn't mean I don't believe your answer. I just mentioned that probably all of us in the room knows that the Nobel Prize laureate, I think one of her paper has just retracted, mainly because she could not reproduce, and it was in Science published, if you like. That is why I'm asking you. Thank you.
You're welcome. But yeah, our results are reproducible. With the same, I'm telling you, if I give you the same clinical samples, we can validate, we can reproduce all of these findings. Any other question?
Hello, everyone. Thank you for coming here. My name is Arwa. I graduated about a year ago. I want to thank the WEP team and the Alumni Office for giving me the opportunity to speak to you today. I was asked to choose a catchy title for my talk, and I came up with this one, Navigating the Complexity of Diseases Genetics for Improved Prediction of Drug Development Success. So you can see there are mainly 3 components in my talk. There is drug development, genetics, and prediction using AI. And nowadays, it is impossible to talk about drug development without discussing the role of AI and genetics to accelerate the process of developing new drugs or repurposing existing ones. So today, I will tell you a little bit about myself, what I did here at KAUST, and how it is relevant to my current work. Okay, so I got my bachelor's degree in computer science from Prince Sultan University in Riyadh. And then I moved here to KAUST, where I joined the Computational Bioscience Research Center, where I got a master's and a PhD under the supervision of Professor Vladimir Bajic. And last October, I was saddened to hear the news of his passing. He was a great mentor and champion, and he supported me in every step of my career, and he made me the person I am now. And for him, I will be forever grateful. Okay, so I'm a computer scientist. I mainly specialize in artificial intelligence, especially applications of machine learning in bioinformatics. And I worked in different topics during my time here at KAUST. I worked on text mining and computational toxicology, in which I developed machine learning models to predict toxicity of chemical compounds, which is important for drug development. And I will tell you a little bit more about this since it's relevant to my current research. So at the beginning, I will explain the drug development pipeline. There are several stages. The first stage is target identification. Target is a molecular in the body, such as a protein, that is associated with the disease. So once the target is identified, you can develop a compound that interacts with the target to produce the desired therapeutic effect. And then after that, you take the target to preclinical testing, in vitro or using animal models, and then you start clinical trials on human. In the first stage, you try it on a small number of the population, and then in phase 3, you test the drug on a larger number of people, and then you move to phase 4 to get the drug approved, and then the last stage is post-approval monitoring after the drug is released to the market. However, this process is lengthy, costly, and it has high attrition rate, meaning the success rate of developing new drugs is very low. And there are several causes of clinical trials failure. One cause is safety. So recent studies show that 25% and 14% of drugs fail phase 2 and phase 3 clinical trials respectively because of safety issues. And in reality, compounds or drugs can fail at any stage of the drug development pipeline because of safety. So it may fail at the beginning, at the preclinical testing, or it may fail even after the drug is released to the market. It could be retracted because of safety concerns. And this is what I worked on for my PhD, is to predict a machine learning model. So to predict at the beginning of the drug development pipeline whether the compound will cause any toxicity effects. However, toxicity or safety is not the only cause of failure. In reality, 24% of drugs fail because of safety issues, but you can see that efficacy is the main cause of failure in drug development pipeline. So 52% of drugs fail because they're not effective. And recent studies show that 48% and 55% of drugs fail in phase 2 and phase 3 clinical trials because of efficacy issues. And in reality, efficacy is the main cause of failure in late stage drug development pipeline. And the reason for that is because efficacy issues may not become apparent until the drug is tested on a large number of people, which is often done in phase 3. So the question is, how can we increase drug development success in later stage clinical trials? And the answer is in genetics, especially the step of target identification. So a recent study published by Matt Nelson showed a strong correlation between human genetic evidence and drug development success, and these results were later on reproduced last year. So drugs with genetic evidence between targets and diseases are more likely to succeed clinically. And if you look at the drug development pipeline, target selection is the first step. If you select the right target, you will increase the chances of drug success. But if you choose the wrong target at the beginning, the drug may fail later on. However, it may take more than a decade to find out if you chose well, because it takes a decade to reach phase 3 clinical trials. And by then, you would have spent billions of dollars and the data you need to choose well is complex and incomplete. So I will tell you about what we are doing at the European Bioinformatics Institute in UK, where I work as a postdoc fellow with the Open Targets team. So Open Targets is a public-private partnership for discovering and for identification and prioritization of therapeutic targets. So it's a partnership between 7 organizations. So there is the ABI and Sanger Institute, and we collaborate with 5 pharmaceutical or biotech farmers. So this is the knowledge cycle of Open Targets. So we tried to find therapeutic targets from different directions. We work on bioinformatics projects to integrate data from public sources. We use AI to generate hypotheses from the integrated data, and then we validate the predictions in the lab. And then we use the data that is generated from the lab, and we release it to the public. I will talk about the data integration part, since this is relevant to my talk. So the Open Targets developed several web resources for finding associations between targets and diseases, and the main platform is the Open Target platforms. So we integrate 7 types of data, mainly genetic association, especially GWAS analysis data, somatic mutations and drugs, especially the results of clinical trial studies, pathways and systems biology, and RNA expression in different tissues. We also use the results of testing on animal models, and we use text mining to extract additional information from scientific literature. So we combine data from more than 20 different resources, and currently we have data on 27,000 targets and 13,000 diseases, and in total we have 6 million associations between targets and diseases. Given the large volume and heterogeneity of the data, we use AI to learn from existing data and therefore predict new associations. So AI is useful to extract information from large volume of data, and it's a good tool to identify relations that may not be easily identified by human insight. And it's useful to propose hypotheses for associations between entities. And in the domain of drug discovery, AI is a powerful tool to accelerate the process of developing new drugs or repurposing existing ones, and it has already been applied at different stages of the drug development pipeline, such as therapeutic target discovery, drug discovery, and safety assessment. And I will focus more on how I used knowledge graph to predict associations between targets and diseases to discover therapeutic targets. So knowledge graphs consist of a group of nodes and edges. So in this case, nodes represent biological entities, such as drugs, diseases, and targets. And edges represents associations between these entities. And the goal of the research is to use existing associations to predict associations between targets and diseases. So knowledge graphs are useful to facilitate systematic integration of data from heterogeneous sources, which is what we have in Open Targets. And it provides a visual representation of relationships. And it's a good way to record how knowledge evolves over time. And in the domain of life sciences, where data is often imbalanced, so in this case, if you have a group of targets and a group of diseases, the number of existing associations or positive associations is usually much smaller than the total number of target disease pairs. And knowledge graphs provide a compact representation of such associations, whereas matrix-based approaches often provide sparse matrices. So this is the graph that I generated from Open Targets data. So we have about 20 different types of nodes, such as genes, anatomy. We use variants, diseases, drugs, and phenotypes, pathways, and so on. So we have 73 million edges in the graph with about 500,000 nodes. And we have about 1.5 million associations between the targets and diseases. And there are about 300 million unknown associations between the target and diseases. And we want to find out among the unknown associations, which one of them could be positive. So I used graph embeddings approaches. So graph embeddings provide feature vectors that provide the feature vectors for each node in the graph, and these feature vectors capture semantics in the graph such that similar nodes have similar feature vectors. And I used the PyTorch Big Graph algorithm or toolbox that is developed by Facebook. So this toolbox mainly takes a list of edges as input, and then it trains a model, and then it generates the features for all nodes in the graph. And then you can use the features to make the predictions whether there is an association between the nodes. So there are different parameters that you have to tune to train the model, such as the scoring function, how to calculate the similarity between the nodes, the relation operation, how to transform the nodes before calculating the similarity, the loss function, which is an optimization function to score positive and negative edges. And also you have to choose the number of dimensions for the nodes, the learning rates, and also the number of epochs. So these are some preliminary results that I got. So you can see, let me just show you. Yeah. So I calculated the hit at 10 and hit at 50 and hit at 100 score. So basically these scores tell you among the testing set how the proportion of edges in the testing set that were scored in the that have scores, for example, of 100 or higher. So you can see in this graph the model achieved about 83% to predict the associations between genes and diseases. And the model is very good in predicting associations between gene expression in specific tissues. And this is a visual representation of the graph embeddings. So you can see that so the genes and the diseases and the drugs are clustered here. And this is the anatomy nodes and the diagnostic nodes. Okay. So in summary, I talked about how to use knowledge graphs for predicting associations between targets and diseases, and also developing machine learning models for therapeutic targets discovery. I talked about the role of genetics in healthcare, mainly in drug development. However, genetics can be used in other applications, such as personalized medicine and preventative medicine and diseases diagnosis. I mainly talked about how we can use genetics to predict efficacy of drugs by finding causal associations between targets and diseases. But genetics can be also used for predicting tractability, for example, meaning whether there is a drug that can interact with the target to produce the desired therapeutic effect. And also, genetics can be used for safety assessment by trying to find associations between the targets and safety risks. I talked about the application or the role of AI in healthcare and how I used knowledge graphs to predict associations between the targets and diseases for target discovery. But in reality, you can use knowledge graphs to predict associations between any pairs of nodes. So you can use it for drug repurposing by predicting associations between drugs and diseases. Or also you can use it for drug discovery by predicting drug targets interaction. AI is not a new field. For example, neural networks were developed back in the 50s, and applications of AI in healthcare are not new either. For example, clinical decision support systems also were developed in the late 50s. However, there are several reasons for the current interest of AI and its applications in healthcare, mainly advancements in compute power and also improvements in algorithms, availability of open source software, which makes it possible even for people who don't have any programming skills to use machine learning to develop predictive models, massive investments by R&D investments by industry, which includes the tech industry, such as Microsoft, IBM, and Google, who also who invest heavily in healthcare research, as well as the pharma and biotech industry, who are also our partners. And availability of big data, which takes me to the next point, which is the benefits of open data in healthcare research. All the data that I use in my research is available publicly. And EBI is one of the main providers in integrating and making this data available publicly by so far developing 57 databases for molecular biology. And also finally, we have to talk about the benefits of public-private partnerships in healthcare. So Open Targets is one of those partnerships that mainly focuses on therapeutic target discovery, but in Europe, for example, there are 70 other public-private partnerships in which the academic sector collaborates with the industry companies to produce scientific research. And yesterday I heard about the Cows to Smart Health initiative to transfer research outcomes from the lab to patients. Finally, this research would not be possible without my team members and their support. Currently, we have vacancies for software developers to develop the platform, but sometimes we also have vacancies for research positions, so keep an eye on this page, and if you are interested, feel free to get in touch, and thank you so much. So you can ask questions. Thank you. Yeah, questions? So you can ask technical questions or even any professional development questions or wherever.
Hi.
Thank you.
Yeah, this actually is a basic question. I'm from outside this field, so
Okay.
actually I don't quite understand the word targets. What does it mean?
Yeah, so a target is a molecule in the body, for example, a protein in the body. So proteins are often encoded by genes, and very often these proteins are associated with the disease. So for some diseases, they are caused because of the activity or the expression of the protein. And then once you find the protein that causes the disease, then you can develop a drug in order to change the behavior of the protein. Okay?
Thank you very much.
You're welcome. Okay, so we have two questions here. Okay. Yeah, so his question was about integrating public data. So I integrated data from about 20 different sources, and his question is whether you should exclude some of those data to get good results. Well, the reason why we integrated a lot of data is because we don't really know exactly which type of data we need to make those predictions. And so you can really actually experiment. Now, it depends on the quality of the data. So for example, the text mining data that we have may not be entirely accurate because it is extracted using text mining. So you may choose to exclude those and mainly focus on data that is from other public sources.
Hi,
Yeah.
Hi Errol. Welcome back.
Thank you.
I just have a question regarding when you talk about the target, because I'm a bit working on a little stuff on genomics. So, and based on what I read, that there's some called linkage equilibrium between the SNPs, and these can have some related with the disease itself. So this software can help us to study this kind of association between these SNPs and help us know how we can map specific SNPs to these diseases.
Oh yeah, yeah.
Because actually I'm interested in this because right now I'm developing some softwares that seeing these kind of association and related is to some deeper technology and high performance computing in order to do some performance optimization.
Yeah, sure. You can definitely use machine learning to find no, I mean this software, no, you cannot really use it because it's mainly to find associations between the genes and diseases. But definitely it is possible to use machine learning for...
through this software?
No, not through this software. Yeah.
Thank you.
You're welcome.
You described this pipeline in which billions of dollars are spent and then no useful drugs, at least for the moment, come out. but one could imagine that some of that data will become useful to a future generation designer. You've described a couple of companies like GSK and Sanofi that collaborate with you. Does that mean they open their failures to the public, or are most companies sort of harboring those billions of dollars of investments so they alone understand what failed as a competitive advantage? How cooperative is the industry in your Open Targets initiative?
So all the data we have is available publicly, but our private partners have their own private data. So GSK and Sanofi do not really publish their own, what they do and their own research. But actually, what we recently developed the Open Targets Validation Lab, which what we use for experimentally validating the predicted targets. And we started doing some experiments, and very soon the results of these experiments will be published. Okay.
Thank you. Very interesting research. I have a question about the graph embedding. Because we are using the big graph developed by Facebook, I want to ask what's your justification of a choice of this one instead of others? Have you tried other methods? For example, graph convolutional networks, so we can use them to get embeddings for the DNA network or genes network. And basically, your question is the link prediction task. So maybe GCN is a good choice.
Yeah, so I chose it mainly because it is suitable for heterogeneous data. So in our graph, we have different types of nodes and also different types of edges. And it is mainly designed for larger scale graphs, and it can run on CPUs. So you don't really need a GPU to run it.
I see. But GCN can be used for heterogeneous networks as well.
Yeah,
it can
sure.
be scalable.
Sure.
It can go very deep.
Sure.
Yeah, but
Yeah.
yeah. Thank you.
Any other questions? All right, thank you.
Wow, it's really great to be back here. I remember my first year when I attended the first WEP program, and I was really excited by all the speakers and programs, and I hope that you're having as much fun as I did in my first year. Thank you all for coming. Just wanted to know, how many of you are students here? That's wonderful. Thank you so much for coming. I'm Faisal Nawab, and I'm currently an assistant professor in the computer science department at the University of California in Santa Cruz. And what I want to share with you today is a question that has been in my mind for almost 15 years now. It's the question of whether we are at the beginning of the next technological revolution. And this question began with me as a teenager. Just graduating from high school, I was going to KFUPM, which is the university at the East Coast here in Saudi. And at the time, I was very fascinated with technology. I read a lot of science fiction books, but I also read a lot of technologists that are predicting what is going to happen in the next decade, in the next few decades. And one that stuck in my mind is Ray Kurzweil, that despite his more controversial ideas about what's going to happen 50 years from now, had a very interesting graph that I always think about. It's that graph to your right there. This graph shows the progression and the rapid progress of technology. And it's really amazing to see that only in the span of 50 years, we have accomplished so much that compared to 2,000 years of development and of technology before that, all that becomes very insignificant compared to what we did in the last 50 years or 100 years. And one consequence out of that is, well, what is going to happen in the next 50 years or the next 100 years? Are we still going to notice or observe the same level of rapid advancement in technology? And I started to imagine all the things, the wonderful things that could happen. And a fascination of mine was in smart cities, in these large-scale deployments, when technology is actually going to be part of our lives. So far, technology has been restricted inside these metal boxes that we need to look at, that we need to interface with. But I really started imagining that we're going to live inside this technological revolution that is going to change everything around us, that is going to take insights from every action that is happening around us and turn it into a useful action that is going to make our lives better. Having grown up in Jeddah and then living most of my adult life in California, one of the biggest problems for me is the problem of traffic. And when I started thinking about smart cities, I started shifting about thinking that, well, maybe once we have all this data collection and all these sensors and cameras around the city, making insights and making actions and crowd control, maybe we can solve that problem. And I want you to now think about the most pressing problem that you're facing, and maybe technology is going to be able to solve it, especially in this context of a revolution in industry, a revolution in smart cities. And with all this excitement, that is when I decided that I need to go to KAUST. That is a very good place for me to try to start my research career and hopefully be part of realizing this dream of mine of the technological revolution. And looking around the computer science department, I started working with Professor Bassem Shahada, who had very interesting work that really aligned well with my long-term goal. He did a lot of edge research, IoT research, all these devices that can be around the world in a smart city, in a large smart factory. How can we coordinate and communicate between all of them? And that was a very daunting challenge that we started tackling here in KAUST when I was a master's student. And while I was finishing my master's program, I started applying for PhD programs. And I remember asking Bassem, what do you think is going to be the best tool, the best research tool for me to realize this goal of getting to that technology revolution? And at the time, it was 2009, and it wasn't that obvious then. But the cloud was still a new thing. Actually, the first Amazon Web Services cloud, which is now the dominant or one of the dominant cloud providers, was only 3 years old. So we didn't really know about the cloud as students back then. But he told me that if you want to solve problems in the future, you want to have impact in these problems, go to the cloud. So I applied widely in the US, and I got offers from around 12 universities. All of them, except one, were offers from networks faculty, because I had this background in networks. And only one group gave me an offer to do work in cloud research. And that group was at UC Santa Barbara. And I took Bassem's advice, and I said, well, I'm gonna trust you on this. I trusted you on many things before, and that was the right decision, so let me try this. And I started working at UC Santa Barbara with a group on cloud services, on building systems for the cloud, and it turned out to be an amazing journey. Because all these companies that are affecting our lives, from search, from social networks, from content and streaming, all these different companies, the main issue and problem that they were facing in the previous decade is, how can I utilize more compute resources? And the cloud was the answer. And it was a very exciting journey because we've observed and helped in building a lot of what's happening in some of these companies. And finally, when I graduated from my PhD, I was lucky enough to get a tenure track position at UC Santa Cruz. And now, beginning as a PI, I thought that now it is time. I've been training for so many years. I've been doing my advisor's work for so many years. Now it's time to bring my dream back and really do something with it. And that dream that I was interested in is to try to utilize technology and computation for this future of integrating humankind with technology. What was really exciting about UC Santa Cruz is that it was only 30 minutes away from Silicon Valley, where all the action is really happening, where all the big impact is happening in computing. And it was a great opportunity for me to go to a lot of different places over the hill, things like HP Labs, Google, RTI, and all these other companies to your right. And I started conversing with them, telling them about what I wanted to do, asking them what are the things that are concerning you. And it turns out that many of these companies are also excited by these opportunities in the edge and thinking that it's actually one step into the future. So I told them, well, now I'm an expert in the cloud. I did 6 years of PhD work on it. Let me try to collaborate with you and help you. And they surprised me with an answer that the cloud is not the right way to support these emerging applications. If you want to do things at the edge, if you wanted to do things that are going to really impact people's lives, you can't really ask a data center that is very far away from you to do the real-time actions and insights that you need in real time. So there was this gap between what the cloud can provide and these applications of the future of technology. So unfortunately, for a few months, all these things that I imagined, I felt that there is a gap that I might not be able to fill. So I started thinking more about it, taking a step back, and I started thinking, what is it that is missing in the current computing paradigm that is needed for us to really get into the next revolution in technology? And it really boils down to two things. The first one is that we need to process more data faster. Once we get into the smart city, industry revolution, and IoT types of applications, the amount of data that is going to be generated is really high. And we need to be able to process all this data. And the other key ingredient in the future of technology is that we need to process this data in real time. A lot of these emerging applications require actions to happen really fast. If you are in an autonomous or self-driving vehicle, you can't really expect to spend hundreds of milliseconds or even seconds to get insights from the cloud. So everything needs to happen in real time. And unfortunately, centralization in general and the cloud were not the answer to these two key ingredients for applications. So I started reflecting on how technology progressed so far, and maybe with this reflection on the past, we will be able to project what is the future and what is the right next step for us as researchers to start doing. So I went all the way back to the 1960s and 70s, and at the time, mainframes were the first type of computing that was supported, where all the resources are with one place at a mainframe, and everyone is connecting to it through a terminal. 10 years after that, in the 1980s, we started having personal computing. And all these PCs had more capabilities, and people would be able to do more on their personal machines. So it started making sense that you don't need to rely on this centralized mainframe, and rather you can just collaborate with your neighbors, with your colleagues, through networks across these PCs. After that, in the 1990s, we started needing to do more in computation. We started observing these .com companies that needed to do search in large amounts of data, companies that needed to keep inventories for a large number of products. So we needed to host more compute and more storage in specialized devices that were called servers that users would be able to connect to. And then in the 2000s, now we started noticing the emergence of other types of applications that required large-scale collaboration between users. Things like peer-to-peer services, Napsters, for those of you who remember that, and also things like content delivery networks. All these things required larger deployments that would work in a collaborative way without having this centralization. And finally, now we're in the era of the cloud, where the type of compute and the type of insights that we need to get from data is so expensive that we need to get all these resources in these large-scale data centers where we have tens of thousands of machines working together to solve our problems. And when we notice this progression of these computing paradigms, we observe that there are two forces that are competing with each other on what is the technology that is dominating in any decade. These two forces are the force of consolidation at one hand, which is trying to put things together so that you can have more throughput, so you can do more with your resources. And the other force is the force of distribution, putting things closer to user so that you can do things in real time. And unfortunately, these two forces go against each other. And as we progress in time, as we see that there is this back and forth between these two paradigms. But the other thing that we also observe is that as we progress in time, each one of these two forces take a different shape. And in particular, it's really the scale of each one of these paradigms or each one of these phases. So if you observe what happened in network machines in the 1980s and compare that with what happened in peer-to-peer networking, the scale is very different. So noticing this pattern in computing, I started asking, are we at a turning point? Are we now at the tip of the cloud phase and moving into the next phase of computing, going back to distribution? And thinking about this problem from this context started making sense, because a lot of the things that I heard from companies and collaborators in Silicon Valley is that there is these challenges in the cloud that cannot be overcome for fundamental reasons. And we observe things like the complexity of IoT projects, where some reports say that 75% of these IoT projects fail, and mainly because of the complexity of these projects. And also the data volumes that we deal with are very huge that cannot really be effectively transferred to the cloud. And finally, there are some fundamental issues too, such as the speed of light. No matter how good your interconnects and your communication is going to be, you're always limited by the speed of light from your device to the data center. And what we've noticed is that for all these different companies around the world that are doing edge databases, they are mostly following the same pattern of computation, which is I'm going to collect the data at the edge from all these IoT or smart devices. I'm going to push it to a middle tier, and then finally I'm going to push it to the cloud. And at the cloud, then I'm going to do all my analysis and insights. And this is not scalable. For those companies, without mentioning names, because it's really embarrassing, to be honest with you, to get insights using this approach, it might take a few hours until you get insights. This is not real-time insights. And this is a clear problem that needs to be solved. So trying to think, well, if we know that we're going to a next phase, well, how does that new phase look like? And in my perspective, I think it's really about three main things. The first one, edge processing. We actually need to do processing at the edge. It's not only for static data. It's not only for these inexpensive operations that are typical now of edge computing, but rather we need to think of the edge as an extension of the cloud, as an extension of computing where we do everything there. The other aspect is decentralization. We can't really have any hierarchy or structure to these edge devices because it's an unpredictable environment. A lot of things can go in an unexpected way. So we really need to build our protocols in a way that is going to be decentralized. And the third one that I think is the most interesting is that, well, what is limiting us from getting to this world where there are edge compute and infrastructure that is around a city, a university campus, and so on? And it's really that it's really very difficult to expect that there is one entity that is going to build all this infrastructure, right? Unlike the cloud, where we know that there is a company that can build a data center, and that's it. Well, how can you expect a company to build an infrastructure that would cover the whole country, for example, right? So this third ingredient is the concept of a contractor, that the devices, the infrastructure is not going to be owned by one single entity, but rather it's going to be a lot of third parties, maybe telecommunication companies, maybe cloud companies, all these different types of companies, each getting a segment of the population, and we try to orchestrate across all of them. And in fact, this has started to happen, where we see that there is a collaboration that was announced last month between Amazon AWS, which is a cloud provider, and Verizon, which is a telecommunication provider, so that Verizon can provide their infrastructure, their telecommunication infrastructure for cloud access, so that we can build applications that can serve people at the edge. So trying to kind of like to summarize how this next phase look like is about having this large-scale deployment around the world and us trying to take these three components and applying them to a system design that is going to solve that problem: edge processing, decentralization, and the concept of contractors. But at the time, we thought that, well, we're in our academic university. Maybe we are sucked in a bubble, and we think that these are the right solutions, but maybe they're not. So we thought, let's do something different. Rather than just thinking inside the lab, let's try to actually build a research prototype and try to see if people are going to use it. So we started working on a prototype that we called Unilog, which is a data infrastructure that is going to be decentralized, that targets these IoT and smart city applications. And we're going to take these three design components, and then we try to reflect them on this prototype. And we were lucky enough to gather a very wonderful team of people from both industry and academia, a lot of them who had been very successful in taking research prototypes that were built inside labs, and then were successful in actually being deployed in real scenarios. And what really motivated us to build that prototype is that although the idea of edge processing and doing things at a large scale was something that was really professed about 20 years ago, or even more than 20 years ago, it's actually only now is the right time for it to be realized. And that is because the recent research that happened in the past 2 years in decentralized computing and other trusted computation areas now enable us to build a large-scale prototype that is going to be both efficient, highly performant, but also trusted, which is one key issue about these edge and IoT deployments. And in a nutshell, our idea of the architecture of Unilog is that we try to make a distinction between the different tiers of computation. So at one level, at the bottom, we have sensors and IoT devices that could be around the city or around the campus, and a group of these sensors would contract with one of the contractors. For example, they could contract with a company like STC or like Amazon AWS to host their data. And typically, that's going to be a company that is close to them. So now the data for this group of sensors or IoT devices is going to be close to the in the data center that is closest to them. And then after that, there's going to be coordinators that are going to enable users to find what are the contractors that contain the relevant data to them and do all the types of manipulations to take a query or a request and then formulate it in a way that is understood by these contractors. And finally, we interface with clients that are used by everyone. It can be interface with something like a web interface, an SAP interface, or anything that is used by the user. And finally, the magic ingredient here to connect everything together is having a blockchain layer, a truly decentralized infrastructure that will enable all these different components to agree to achieve consensus in a trusted way. And what is interesting is that it's not only about coordination and consensus. What is interesting about blockchain is that it also integrates a compensation mechanism, so that when I'm trying to contract with a contractor, within the same distribution and coordination framework, I can exchange payments and compensations. And I can also dispute if someone did something wrong. So all these new advancements in blockchain is really the enabler of a lot of how we connect all these different components together in a decentralized way. So to take a quick example of how this can happen, if we take these sensors that are at the bottom and the contractors that are on top of it, all this data is going to move from the sensors to the contractors, and we get queries from users like, let's list the water meters that are showing a so-and-so increase in usage, and through knowing what are the contractors that hold this data, we can send that query and get the information that we want. And this can happen for all different kinds of queries as long as we know where the relevant storage and contractor devices are. So we took and built that prototype, and luckily we've enjoyed very great collaborations now with these companies that are deploying our POC in their applications. And to give you a sense of how one of these look like, this is one of our deployments in a smart city application. So that is a company that does fiber optics in a city in California. And what you observe here is that there is these sensors that are around the city that are collecting all the data. And then we have this unified interface that is going to only communicate with the contractors that are relevant to any query that we are now issuing through this interface. For example, if you see these plots, these are the plots of the readings from these sensors that are around the city. So to conclude, the next technology revolution, I think that it's looming. I think that it's happening right now. And I think a key ingredient in achieving this technology revolution is through designing a decentralized data infrastructure that is going to get all these three design principles and have them in one solution. And hopefully this is going to enable us to achieve these dreams and goals of the industry revolution and a truly smart city. And before I conclude, I'm going to thank the students and collaborators that are actually doing this wonderful work. I thank the undergrads and graduate students at UC Santa Cruz who've started building a prototype of many of these solutions. And I thank the PhD students that are tackling a lot of these daunting research challenges in the process. And finally, a very quick plug about an event that we're holding at Santa Cruz. It's a symposium about the foundations and applications of blockchain. It's the third year that we're hosting this event. For 2 years, it was held at the University of Southern California, and it's our first time holding it at UC Santa Cruz. We're very excited by it. We have a collection of very excellent keynote speakers. The deadline for submissions is still next month. So if you're working in a related area, I'd love to see your submission there. And of course, let me know if you need any help in the process. Well, thank you so much. Okay, so now it's time for the game of cube rolling. Okay.
Yes, I'm so glad you're close, so I don't have to throw it.
Yeah, thanks for your nice talk. But for blockchain, the proof of labor, the proof of stake, that will happen in the edge. That one question, and then what do you think about NEOM and the Chaos Smart Campus?
These are very excellent questions. So for the first question about blockchain, so I intentionally didn't say anything specific about blockchain because there are properties of blockchain that are going to be needed for these types of applications. But it really depends on the constraints that you have in your organization for the type of blockchain and the type of proof of stake or proof of work that you need to deploy. But if we are doing something like a proof of work or a proof of stake technology, this is going to happen in a decentralized way. So there is the chance that it's going to be a very expensive process. So what we try to do from our end is that we limit our reliance on the blockchain network only for the metadata and configuration tasks. But all the processing, all the requests happen off-chain. Yeah, but that's a very excellent question. As for NEOM and the other smart cities, so you said that there is also an initiative here in Chaos that I think I started hearing in my visit a couple of days ago. I think this is very wonderful. I think really now is the right time to invest in these advances. And I think in NEOM, in this large scale of a city, and also at the scale of a university campus, I think both there is a lot of great opportunities. If someone here knows more about it, I would love to talk more about that. Yeah. Any other questions?
Thank you, Faisal, very much for your interesting talk. And actually, it is very impressive to see how humankind evolving from centralized to decentralized movements over time. And actually, when we look at today's situation, that's not going to be enabled tens of years ago when they were using centralized solutions because of the advancement of technology today. And actually, what's always concerning also people is about privacy of their information. How do you think moving from centralized to decentralized or the other way around would impact the privacy?
This is a wonderful question, and I'm really happy to hear it, because this is one of the most challenging aspects that we're trying to deal with. And I think this gets back to what I mentioned a few slides ago, which is now we are at the right time to tackle this problem. And that is because a lot of the privacy and security challenges, we didn't know how to solve them a few years ago. And now we have the technology and the research to do this efficiently. So for example, for the issue of, let's say, secure computation. Until recently, we didn't know how to enable an untrusted device to do computations in a trusted way in an efficient time. But only in the last few years, we started having hardware, things like trusted execution environments, but also software concepts that is going to enable an untrusted entity to provide a proof of the authenticity of their response. Of course, this applies to all the different challenges in security and privacy, and I'd love to talk more about that after the lecture. Yeah. Great. Okay, so I'm going to stick around, by the way, after the series, if you need to talk or discuss or just say hi. So yeah, hope to catch up with all of you.
Hola, hola. Yes, it works. Well, do I need to do this? Hello? Hello? Yeah, I think here it's gonna work. So yeah, hello everyone. And so I'm the last speaker of the day. And when they asked me to give a title to my presentation, well, I was thinking like, yes, I'm gonna talk about collagen design, But as I'm gonna talk also a little bit about what I did here in KAUST, I decided to give a more ample title, and well, it's more like an adventure. So yeah, you're gonna hear a bit of my history. And actually, I'm gonna start talking about who is myself. Well, yeah, my name is Eduardo Gorron, and I'm from Colombia. And I guess many of you don't know where is Colombia, or probably have heard just a few things about it. So it's a country here in South America, in the north part of South America. We are in the equator. Some people tend to think, oh yes, we are a tropical country, so we are always in shorts and very hot. But actually, my city, it's in the top of a mountain. It's 2,600 meters high, so it's actually quite cold. We have an average of 17 the entire year. The entire year, can you imagine? Like all the time freezing. But anyway. And I did my biology and microbiology undergrad there in Colombia, and it was in 2008 and 2009 that the KAUST delegation arrived to my university and started to promote KAUST. In 2009, I got in touch with the delegation that went to visit, and it was very interesting to see that, well, I was apparently the only one that has the GPA that the university was asking at that time to be accepted in here. So yeah, that's how it started. And then in 2010, I came here to do the Master in Chemical and Biological Engineering, which was a very great opportunity to work with different professors, but also was a great opportunity to travel, as much of you maybe know, right? And basically, I knew the world by being here in KAUST. Yeah, something that you cannot do in Latin America. But this talk is mostly about science, so I'm going to talk very briefly what I did here in KAUST, what was my master work about. I work in the Water Desalination and Reuse Center under Professor Pascal Saikali. And basically, we were working with what are called the microbial electrolysis cells. So to make it short, when we eat something, our carbon source of our cells is glucose. The process of respiration basically consists of extract the electrons out of this glucose and transfer to oxygen. And we get the energy to do all the marvelous things we can make by doing that electron transfer. But some bacteria are able to do something called extracellular electron transfer, meaning that you can take those electrons to the outside of the cell. And in the particular case of bacteria like Shigella or Eubacter, you can transfer them to metals outside. So you can say like, okay, this is wonderful. You can actually have electrical current, right? And that's the principle of these reactors. So on the left side of this reactor, of this image, we're going to have the place in which you grow the bacteria. They use the carbon source. They extract the electrons out of it, and you can force the electrons to go through a circuit and go to the other side. You may use this to produce electricity itself, but you can also use the electrons to produce other things, like in this case, hydrogen, which is the main objective of these cells. And this actually comes as a very green, sustainable technology because you are using wastewater to feed the reactor. So you're using waste and produce energy, which sounds actually wonderful. However, always comes with technical challenges. And the thing is that these bacteria use mostly acetate as the main carbon source. But what happens with real wastewater? Real wastewater is like a mix of random stuff, right? A lot of different carbon sources. So it's actually gonna work with the real wastewater. And also, these reactors operate in anaerobic fashion. And when you talk about anaerobic fashion, the main metabolic activity you have there is methanogenesis. So what we wanted to do here in the work here that I did for my master's was to compare the operation of different reactors with propionate, which is a more complex carbon source, and in different concentrations, and also inhibiting or not the methanogens. And of course, there are a lot of results, but I'm gonna just focus on some of them. And on the graph in columns, we can find out that actually increasing the concentration of this propionate resulted in not very good performance of the reactors, meaning that part of the electrons that were coming out of it were going actually somewhere else and not to the production of electricity and hydrogen. But when we analyzed the microbial communities that were living there, we didn't find major difference when we inhibit or not inhibit the methanogens. So from that, it comes actually a paper in which we were proposing different alternative pathways that the electrons could be taken inside these reactors where we're feeding them with propionate. Also after that, then I work here in SABIC as an engineer. And I shift to work with microalgae. And you should know that Saudi Arabia has a very special environment to grow microalgae because we have a lot of land and a lot of water that cannot be used for anything else. But the reality is that we have a lot of sunlight, and the sunlight could be very detrimental for this algae because it's very intense, and the seawater is also very salty. So that's actually a challenge. So while I was working on this was trying to identify a strain that could survive those extreme conditions and that we could engineer to produce compounds that could be interesting. Well, but what happened then afterwards? Well, the project with SABIC didn't really take off very long, and I ended up without jobs, so then the real life comes. And you should know that, yeah, we're basically here in KAUST, we are kind of protected. We are actually having a very good life inside KAUST, and the world outside is way more tough than what you face here. So it came actually a time of unemployment. It came a time of when I was looking for a job without being very successful, to be honest. I went back to Colombia, I went back to my family, was nice in that sense, like meet again my family after a long time here, 4 years here in KAUST, but was also to face again the problems that we have in Latin America implementing these technologies. And Latin America is still not prepared to overtake many of the nice technologies that we have the chance to work here in KAUST. But then I say, okay, we still want to keep the contact with science, and then I become a university lecturer, and I become a very successful university lecturer. I was lecturing at some point in 40 different universities in Colombia, talking about science, talking about all these nice technology, and try to inspire new generations of Colombians to go abroad and probably follow the steps that I have done. But anyway, just lecturing keeps you disconnected from what is actually really happening in science. And then I say, okay, probably it's the time to do a PhD because I haven't done a PhD so far. I'm just doing it now. And for different reasons, I ended up in Australia, which probably when you think about Australia, you think about the crazy animals that are there, the kangaroos, the koalas, or the very nice beaches that are there, or that there are very sexy people in the beach. But probably we don't realize that in there we have one of the top universities in biotechnology. At the moment, number 9 in the world in biotechnology. And this is the place when I move to start to do my PhD, and probably actually my talk starts here, when I shift completely the topic of environmental microbiology towards something more biomedical and engineering collagen, which is my PhD project about. When we talk about the clinical problems that our world faces, we think about the typical things: cancer, diabetes, obesity. Yes, those are big problems, but we haven't realized that wound care, the treatment of wounds, is the 3rd major cause of expenses of the health system all over the world. Those are numbers from UK. Australia is not an exception either. We have more than 400,000 people affecting of what are called chronic wounds, which are wounds that don't follow the normal healing process. And these people are expecting for a solution, for something to help them to treat the wounds. And Middle East is not an exception either. Those numbers are talking us only about diabetic food. And we have around 18% of people in Saudi Arabia suffering of diabetes, and from then 25% suffering of diabetic food, which is one of the most serious chronic wounds. And then because of that, we have a market size of around $70 million by 2018, combining Saudi Arabia and Emirates only. So that's something that cannot be ignored. So that's a very serious problem in terms of health. And then collagen grafts could come as a treatment for these chronic wounds. So there are already alternatives in the market, but they are based mostly on the employment of synthetic materials. But actually, collagen could be the most promising material to treat wounds. So you may ask, why? Why collagen could be important in this direction? Well, collagen is actually the main component of our skin, not to tell about other connective tissues like cartilage or bones. And why is that? Because it's the main component in terms of the fibers that are present in the skin, explaining the structural support that our skin has, and also giving tensile strength and flexibility to it. Also, collagen, when we zoom in these fibers present in our connective tissues, we find out that collagen has a lot of regions that are able to bind different components of the extracellular matrix, such as the fibroblasts, such as elastin, hyaluronic acid, and many other compounds by what are called the collagen functional domains, which I'm working a lot now. And when you zoom these fibers even more, the reason of the stability lies in the protein structure that this comes, that is a triple helix stabilized by amino acid called hydroxyproline. Well, collagen, it's already available in the market. It's not a new product. But the problem is that most of it is coming from animal sources at the moment, or from dead people, but that's more complicated. From animal sources, mostly from cows and from pigs. And that sounds okay because they are cheap, but there are several problems. One of them is the rejection. Come on, imagine you have a piece of cow in your arm. Your body is gonna reject it very quickly. And this is a serious problem in medical research. Also, there is a high potential of carrying infectious material from these animals. And finally, you have very limited engineering of the material. So animal collagen cannot be tailored and optimized for specific applications. All these problems may be solved by the use of recombinant collagen, meaning taking the genes of human collagen and expressing them into a protein expression system. And that's what I'm working in. So trying to produce collagen that could be all engineered for wound healing, but also that can be produced at a scale important for industrial production. I'm going to talk first about the nicer part, which is the engineering. And then the first question is how we can design a functionally improved collagen. How can we engineer the collagen to make it better for wound healing? And the first thing that you may have in your mind for this is that usually when you hear about collagen, you may think about these pills that they sell in the beauty salon, right, or the beauty shops. And you take the pill and you have the injection, and wow, now you have a super skin. But that's an oversimplification of the problem. And in our bodies, actually, collagen comprises 28 different proteins. It's a very complicated family of proteins. And that's the first thing that we need to break in terms of the dogma of collagen production. There are many different types of collagen with different roles according to the tissue. So my first idea was like, okay, why we not instead of playing with one collagen, we try to make a collagen that combines the best of the different types of collagens. And then I'm coming with this engineered collagen optimized for wound healing. What the bar that is showing there is an oversimplification of the protein structure of collagen, and the numbers in italic, in Roman numbers, sorry, they represent the type of collagens from which these regions are coming from. So as you see, I'm using elements that comes from collagens type 2, 3, and 7 mostly. Which elements I'm using? Well, I'm using an element called von Willebrand factor that is very well known to attract platelets very strongly. And this is very important because the platelets start the coagulation cascade and they actually start the wound healing process. Also, I'm adding elements of fibronectin that can attract still the platelets, and a fibrin protein important during the first stages of clot formation. I'm attracting using heparin that also attracts platelets and different elements of the extracellular matrix, and also integrins. Integrins are proteins present in many different cell types and help the cells to anchor different components of our body. And in particular, fibroblasts, which are the cells specialized in produce collagen, they have special types of integrin that can bind to collagen. So if we have extra number of integrin binding sites in our collagen, we may stimulate the binding of fibroblasts and then stimulate the production of collagen during the process of wound healing. Another challenge is the production of fibrillar collagen. So if you remember the image that I showed you, you see very nice fibers of collagen, which are very complex structures. But actually, because of that, then the research has been focused on trying to produce those fibers. In the reality, those fibers has not been produced using recombinant proteins. It has been really, really, really challenging. And when you check the literature, it's not very clear what are the key elements that can make this shape to form. So then I say, okay, let's just stop thinking about fibrillar collagen, and why it has to be fibrillar? Why we not instead go to collagens that form other geometries like networks or hexagons that exist in nature that are very well characterized as for collagen that I'm showing there that makes a network. And I say, okay, instead of trying to make fibers of collagen, let's make networks of collagen. And that's another engineering element that I'm introducing in this case. So how does this work? So in the sides of the collagen that I'm engineering, we are going to have what is called the 7S domain, which is a recognition element of collagen 4 that allows the collagen 4 to make tetrameric structures. And then you can have the network. But then I say, okay, let's go one step forward, and let's combine it with another type of engineered collagen that doesn't have this tetrameric element in one extreme, but instead has the von Willebrand factor that I was mentioning you before. So you will have this type of tetrameric structures formed, and the prediction is that when you combine when the original construction alone, you will have a branch network of protein that is exposing a lot of von Willebrand and should attract a lot of platelets and factors to treat the wound. But then we are going back to other problem. All of this sounds very nice, but we need to make it, right? And then comes the problem, how we are gonna produce actually this protein? Well, actually, there is already a disease called fibrosis. In this disease, the fibroblast is overproducing collagen. The collagen production has gone out of control. The degradation of collagen is reduced, and the synthesis is exceeded, and then you will have patches of collagen in your tissues. Can we mimic this phenomena? Well, that's what I'm trying to do. So the pathway that we have in there is a cellular pathway called AKT-mTORC, which has been identified to be overexpressed in fibrosis. And in particular, the protein LARP6 has been associated to the specific regulation of collagen 1 mRNA, stabilizing it and helping to overproduce it. So what I'm proposing is to add this recognition element to the collagen production, to the collagen structure that I'm proposing, and then have this element in a combination with other series of mutations that have been already described in literature that are helping the fibroblasts to produce collagen in high amounts in the fibrotic scheme. So that's another part of my project, so in the engineering of this double mutant expression system. All of this sounds very nice, but you should know that in science, nothing is how it looks, right? Actually, we have faced a lot of problems trying to make this production. So we tried first what is called the XP293. XP293 is a very well-established mammalian protein expression system to produce collagen, but with the yields that we have observed have been very low and have been below the detection levels of our techniques. Then we say, okay, let's go back to the literature and try yeast. Yeast has been described also to produce collagen. However, the formation of the helix has been a very big challenge also, and we have not been able to detect collagen with the right conformation. Then, okay, they say, okay, let's try to engineer the fibroblast, which is what we are trying to do now. However, we know already that this comes with a lot of challenges. It will be very hard to scale up because there are cells that are adherent, meaning they grow in the walls. That will be hard to scale up. Previous reports already say that we are going to get still very low amounts of protein. But anyway, we are going to try it. And we still need to get more information about this environment of fibrosis that helps the cells to overproduce produce collagen. So what are the alternatives that we are already envisioned to work with? Well, collagen is not a hallmark of humans. Collagen is a hallmark of animals. All different animals have collagen, and you can explore the animal kingdom trying to find alternatives of collagen. And one of them is this sawfly that still contains a type of collagen that has a very interesting stability property in terms of temperature. Also, analyzing bacteria, you might find a protein that looks like collagen. It's a small version of collagen, and that has been already produced in high yields in bacteria. So actually, both of them have already been produced in bacteria successfully, and they don't need this hydroxyproline, this special amino acid, to create a stable triple helixes. So they are very promising. The main disadvantage that this comes with is that they are not human collagen, right? They are proteins that are different to the human one, so you may face immune response. However, some authors say that probably this will not be a major problem because collagen is not as immunogenic, but that's something that we need to test. So actually what I'm proposing now is to keep all these design elements and play with our and humanize insect collagen, try to produce insect collagens with the human elements, trying to avoid this hydroxyproline, this helical formation step. Also making a shorter collagen that can be produced in smaller scales and can be mixed with hydrogels. So trying to not have your wound dressing only made of collagen, but mixing it with already synthetic materials to that can help to give support to the cells that are already present in the patient and increase the treatment and the treatment. Also, well, there are isolated reports about production of collagen in tobacco that they are already in clinical trials. And well, there is the alternative then to switch to tobacco and then stop working with human cells and work with tobacco cells. But then finding that I had recently is that algae can actually produce hydroxyproline as well. So why do we not try to produce collagen in algae? And that's the next step that I'm gonna take it. And the vision that I have in this is trying to produce hybrid collagens that don't exist in nature and that have different mixes of different collagens present in our body. This is not an isolated work. This is in collaboration with Professor Fiona Woods, which is a very recognized clinical surgeon in Australia. And connecting with the topic of personalized medicine, her proposal is to take the cells out of the patient, cultivate them outside, and re-implant them in the patient itself. So using the same cells of the patient to enhance the wound healing. And the part in which I play the role is to create the wound dressing and particularly to create this type of collagen so these cells can be recruited to that point and we can enhance the wound healing in different patients. So I'm going to skip this. So actually, I just want to acknowledge the group that I'm working in University of Queensland in the protein expression facility, and also the group of Alan Rowan in the Australian Institute of Biotechnology in University of Queensland in Brisbane, Australia. And with that, I'm happy to receive questions and say thanks for your attention. Shukran. So Leah has the cube in case someone has some questions that you want to make.
All of your illustrations, perhaps because they're on a slide, showed two-dimensional structures. And indeed, the applications looked like they were also layered in two-dimensional structures. But are the collagens actually, are all those like tetramers coplanar like that, or can't they be, sort of 3D masses?
Well, actually, the collagen in our body, and in particular the most abundant types of collagen, they make fibers. So what you have is the collagen monomer. 3 of them associate to make the first helix, and then those helixes assemble one over the other to make a fiber. And those fibers are crossing our different connective tissues. The structure in our tissues, we can find that these fibers are actually doing kind of different, like a mesh structure, mostly supported by elastin and hyaluronic acid, which are the other main components of the skin, at least. Yes.
So when you try to grow them artificially, do they naturally grow in these planar structures? I mean, they're fundamentally 1D fibers, but it looked like all the applications you had were, two-dimensional. You might wrap them around a wound or whatever. But
yes. I think for sake of time, I oversimplified many of the things here. I mean, but yes, actually, how it's supposed to work this wound dressing. So you will have the dressing, which will be a two-dimensional thing, only with a layer of collagen in there. You will attract the cells on it to grow, and they will start to produce a more three-dimensional thing underneath. That will be the strategy. And so actually, I didn't want to go into much detail, but the idea is to attract cells that can produce collagen 4. 4 makes a network that serves as a support for other cells to start to make the fibers underneath the collagen 4. Yes, that's basically the idea. But then you will have, actually in the skin, you will have different layers of different types of collagen arcing or doing networks or doing fibers, depending on what type of collagen you are talking about.
Hi. So I have a question. I think it's the slide you skipped, which was titled "What I Learned from KAUST." I think that's more. Many people here would be interested in learning about that, your experience and what you learned, and how can they apply it to themselves.
Right. Yes. I was looking at the time, and I say, well, I will not stop here. But actually, I think something that I learned that I didn't have the chance to learn in Latin America is the importance of being multidisciplinary nowadays. I think in Latin America, we still have this vision that if you are a biologist, you only work with animals and you only think in terms of biology and DNA, and that's it, and that's the end of your world. Or if you are engineer, computer engineer, then you only work with computer, and whatever comes out of computer is no, that's not my world, and I'm not gonna even touch it. But the reality is that we have a very interconnected world nowadays. And that's probably one of the main lessons I got here from KAUST, in which all the projects are very collaborative. You collaborate with a lot of centers at the same time. You have to collaborate. Me as a biologist, I had to collaborate here with engineers, with electrical engineers, with computer engineers, and learn from each other. And I think that's something you shouldn't miss here. You should miss that opportunity. In particular, I think that KAUST made a very huge effort for every one of the batches that were coming to make us together, and was very enriching to go and have lunch one day with the group of the computer engineers and talk about what they are doing in their projects very crazily, and then with the guys that then they are doing circuits and trying to learn, okay, what are you trying to do in your circuit, and then the guys of membranes are like, how this membrane is working. And combined to the, let's say, your normal academic formation, I think that was another very enriching part of my experience in KAUST, and that's a part that you should take advantage while you are here. Yeah, well, if no more questions, then I will give the word to Leah, I guess. Thank you so much.
So I'm very happy and excited to welcome all our alumni. I mean, when people ask me what's the outcome of KAUST, I mean, people expect me to tell them about high citations, high impact factor that we publish in, high visibility professors, highly cited professors. But in reality, the best and the most important outcome of KAUST, or a product of KAUST, are the alumni, are the students. I mean, so please, join me in thanking them for their experience and the knowledge that they shared with us about their journey outside KAUST and during KAUST. So that. Another thing that's important, I guess Faisal is the only one who enjoyed the free food. So that's something that you guys are not going to be having anytime soon, except during events. So you'll have to come to an event like this, listen to a talk, and then you can get free food. But he had the luxury of having free food for I think 4 or 5 months. So that's one thing. I'm really touched by two of the alumni who actually mentioned one of our colleagues, Professor Bajik. I mean, he was a founding faculty at KAUST. He did a lot to build KAUST from day one. He was here, I believe, in March 2009. So this is way before KAUST started, and he played an instrumental role in building this institution. So really, we really owe him a big debt, and I'm very glad that they owe him this debt, and they actually acknowledge his contribution to the university. One thing that I think one of them touched upon, and I really can't emphasize it more, is the value of networking. After you leave KAUST, you will be left with knowledge that you learned from KAUST, but you're going to have with you a network of people that you met and you interacted with, and they will be the first people who will hand you support. They will help you, because if they help you, they will know that you will help them later, and this network is going to go a long way with you. So please, do not sit in your lab working at your desk or inside your lab and being isolated from other people. Reach out to different people from different labs, different groups, get to know them, get to interact with them, because you never know what's going to happen in the future. You might end up deciding, oh, I like to go to Australia. Then here is someone who's in Australia, he likes KAUST, he knows me, he wants to promote KAUST because KAUST is something that he's proud of. So please try to make this opportunity to interact with people and take advantage of networking. They are our ambassadors right now. You are going to be our ambassadors in the future. So we owe you a lot of gratitude. We will equip you with all the knowledge we can, and we'll help you and support you, because your success is our success. With that, I thank you all, and I wish you a very good day. Oh, one last thing, I'd like the speakers to come